All right, hi there AP Bio students. I've got some more gene regulation material for you for our final needed unit of AP Bio. It is, yikes, Thursday? Sure, March 26th. Hard to tell what day of the week it is when you don't leave the house. But I'm going to go through just some basic introduction, and then I'm going to go into our Operon graphic organizer. So you'll be able to find this filled out on Schoology, and as always, I love to make changes, so you could find those as I go through the video. Got cat hair on this one. Nice. All right, so for prokaryotic gene expression, we're talking about bacteria, okay? Bacteria are very simple. They've got their cell wall, cell membrane, free-floating circular DNA, so their DNA is in ring-like structures. They may have just one major one and maybe a few other small ones. I'll explain why in our next week's material. They don't have linear chromosomes that look like this for what we would think of in eukaryotes, what we've been talking about with meiosis. That is not going to be found in bacteria. But they do have ways to make things more efficient. They may be pretty simple, but they've been around longer than any other form of life. So they have some really efficient processes to help them conserve energy, resources, maximize efficiency when certain resources are available, or how to make them themselves. So just like you and I and the goldfish and the cherry blossom that I have here on the table, they're all using the same mechanisms for transcription and translation. You may recall, or let me go back in time, here on the transcription graphic organizer from last week, the process here in a bacteria cell, DNA's there, make the RNA, ribosomes are immediately available. But eukaryotic cell have a couple more steps that adds to our complexity, like the RNA processing, and the fact that we can compartmentalize transcription and RNA processing in the nucleus and separate it from translation. Bacteria can't do that. So they have to be able to find a way in their studio apartment that is their cell to find ways to turn things on and off. They are going to utilize the presence or absence of certain molecules or substances to then help them regulate turning the genes on and inducing them, or turning them off and repressing them. So just because they're a tiny little cell with not a lot of stuff in it doesn't mean they cannot regulate what needs to be happening. So we're going to be talking about operons, and in the Bozeman Science review video that we've got in the online resources, he uses the acronym PROG. Not like the city, which is P-R-A something, but PROG is in P-R-O-G, and I love it. I did a typo right here at the end. But an operon is a set of pieces of DNA and components here. We're going to have a promoter, a repressor, an operator, and some gene or genes. So all of these are going to be working together collectively as an operon, and I'm going to go through for you first an inducible operon and then a repressible operon, and you'll see that they both of them carry all four of these components. So here we go. When you're filling out this graphic organizer, let's see if that's so far, you may notice it's got like two sides to this, and then when it got scanned, there is a reason why there are these pieces on the back here. So you're going to fold this side down, and then this has the information and the story about what's going on here on the inducible operon. And then flipping it over, fold the left side down, the repressible operon, so now you can see the details about what is happening in these pictures and why it's on and off. So I'm going to be starting with the inducible operon, our lac operon. So this is, first of all, nothing to do with lactose intolerance in humans. Let me adjust here, I'll hold that down. Lactose intolerance has to do with when mammals who are raised on milk in early development, they need to be able to break down the lactose sugar that is in milk. But often mammals are not continuing to eat milk into adulthood. So their body as a eukaryote is like, oh, you don't have this around anymore. I don't really need to be producing lactase enzyme, which is different than the enzyme that we'll be talking about here in the bacteria. So that's why it's a totally different process. If you are lactose intolerant, you eat dairy and you end up having a lot of digestive issues because you can't fully break it down because your body hasn't been doing it for a long time. But with bacteria, we're probably talking about bacteria that are in your gut, and if you do eat dairy, that's now a present food source for them, right? And if that's the only thing in the stomach right now and there isn't any glucose, well, then they got to do something with the lactose and be able to break it down. Otherwise, they're going to starve themselves. So this is about bacteria utilizing available food resources for themselves, okay? So if there is no lactose, let me move closer here. If there is no lactose in the environment where this bacteria cell is living, then the operon or the lac operon, okay, LAC, lac operon, is off, okay? No lactose sugar is present. Transcription of this beta-galactosidase gene, which is the gene that will help the bacteria to break down lactose, it doesn't need it, okay? So let's look at the components here that are going to be needed. I said that we are going to have PROG, P-R-O-G, promoter. There's two different promoters. There's a promoter in front of the regulatory gene, which makes our repressor, okay? And there's also a promoter in front of the gene for the gene in question that we're talking about, okay? So the repressor, here is also RNA polymerase, very similar to the triangle that was drawn on with our transcription notes. So RNA polymerase, it's going to bind to the promoter and just move along and produce this repressor protein from the regulatory gene. There's nothing stopping it from doing that, and it in fact is always doing that, and this protein is always being transcribed and translated. So the form that it shows up in is active. You can see its face, like, I'm ready to get to work, and it's not asleep, it's ready to go. And this active repressor protein has a specific shape. Where's my yellow? There it is. Looking at its shape here, these like three little bumps, there's a reason it has that shape because it matches the shape of what we call the operator. So the operator is another region of DNA nearby, right? But not actually related here, right? It's got its own promoter again. So this is the lac promoter, which is going to be related to the lac operon. The operator, which is kind of the on or off switch here. If the active repressor protein is in its active form and can bind to the operator, RNA polymerase gets blocked. It's not able to continue smoothly along the DNA chain to be able to transcribe this gene. So we don't get transcription of the beta-galactosidase enzyme, which would be breaking down lactose. There is no lactose present. We don't need that gene. So this is a way to turn transcription off. Now, if we want to induce, that transcription, we got to turn things on. There we go. Okay. So let's say this bacteria's environment doesn't have glucose available currently, but it's got lactose sugar, okay? Lactose sugar is actually a glucose and a galactose 5 or 6 ring sugar, don't remember, but they're connected to each other as a disaccharide, okay? So if that disaccharide is present, that lactose sugar actually has a binding relationship with the active repressor protein, again, getting made in its active form. But if it does that, it's going to change its shape. And so now I want to get red. Boop, boop, boop. So we've changed the shape, and now it's in its inactive form, and it cannot bind and fit to that region on the DNA, right? It's not going to block that operator. So RNA polymerase is going right along and will go ahead and transcribe and translate the beta-galactosidase gene, and we get beta-galactosidase enzyme. And look at this one, has the right shape to bind to lactose sugars and be able to break them apart in a catabolic reaction, okay? So beta-galactosidase enzyme is the protein that will break down lactose into glucose and galactose. If you were measuring this in an experiment, you could measure the production of glucose, right? If that is going up and up and up, you know that this process is active. If you have lactose sugar, it's going to start to get lower and lower and lower as we get more and more glucose present, okay? So some of the things I've got on the side here to summarize inducible operons. This is what the bacteria might be saying. So some of it got cut off, so I'll go ahead and read it aloud. Hey, this food I need to break down is in my environment. I better induce transcription of the genes that produce the proteins to help me digest that food. So basically, if lactose is there, I better induce transcription of the proteins that help me deal with lactose. Okay, that's what we mean by inducible. So typically, inducible operons are related to catabolic reactions, reactions where we're going to break down material, there we go, and break them down into smaller parts, digestive reactions. Not necessarily us human digestion, but this is probably how bacteria are helping us to digest certain foods in our stomach as well. It could also be like the kind of bacteria you would imagine in like that help us make yogurt and dairy and cheese and things like that, not necessarily in another organism. So when lactose sugar is present, or it's the only resource available, right? If you have to do all of this every time lactose is there, that's not great. If you've got glucose available as a bacteria, go for it. You don't even need to do this process. But if lactose is suddenly your only resource or it's more plentiful than your other resources, now it's time to turn this on and start utilizing that food resource. So the cell's genetic mechanisms will induce transcription of the genes that help to digest that sugar. If lactose is not present, no need to waste energy on forming those proteins, right? So if it's not present, we're currently repressed and we don't have our lac genes turned on. Our repressor is active and blocking transcription. So lac operon is one example. Another example that is pretty popular that they love to throw you a curveball on the AP test is the arabinose operon. So it's still just another sugar. Don't forget that lactose, OSE ends in, or sugars end in OSE, and here's arabinose, that's another one. It's just another kind of sugar. But here's the little phrase like, X is present, operon is on. That means it's an inducible operon. So that statement, if your molecule in question is present, and in that case the operon is on, it would be classified as an inducible operon. Okay? Alternatively, let's start at the bottom here. If your molecule in question X is present and the operon is off, that's going to be the repressible operon, so the opposite situation here. So let's go to the top here for the trip operon, okay? For tryptophan operon, tryptophan is an amino acid, which is a building block of proteins. You've got to have enough tryptophan to be able to make the proteins you need. So if there's plenty of tryptophan already present in the cell of this bacteria, then we don't need to be making more tryptophan. We've got it, right? So let's go ahead and turn off the transcription of the genes that help us to do tryptophan manufacturing, okay? So again, we've got a regulatory gene that makes a repressor protein, but it's initially made in an inactive state. So this is not the right shape to bind to the operator, right? That's got 3 bumps on it. But that's in a repressible operon. It's initially made in the inactive form of a repressor protein, but that tryptophan is going to bind to it as a co-repressor, okay? Something else needs to be there in order to repress. So now we've got active form, and that active repressor protein with tryptophan bound can bind to the operator, okay? So we still have a trip promoter that is in front of all of these genes here. In this example, it's not just one gene, but like a series of genes that come together that basically become the series of enzymes that will do the several step process to make tryptophan. It's not a one step process. So here's a way that bacteria cells have become efficient by just like, hey, I'm going to need enzyme A, B, C, D, E. Okay, let's just put them all together because whenever we're working with them, we use them all, right? So there's one promoter in front of it. Operator with an active repressor is bound and blocked, so RNA polymerase is not able to move forward. It is blocked, okay? But that inactive repressor protein only becomes active when tryptophan is there. So it takes that binding to change its shape into the active form. So keep in mind, like from our biochemistry unit early in the year, binding of something can change the shape of something else, like a phosphate, in this case, a tryptophan, okay? But now tryptophan is depleted. Let's say for this bacteria, there isn't enough protein or other amino acids in the environment. It's depleted its supply. You don't see any tryptophan present. So this inactive repressor protein with the wrong shape stays inactive, okay? There's no tryptophan to change its shape, so it's not going to be able to bind to the operon. And, well, there you go. RNA polymerase is free to move forward and go forth and do transcription of the rest of the proteins here that are going to be needed to help us manufacture tryptophan, us being the bacteria cell. So it's going to have protein E, D, C, B, A. The names of these are not important, but small molecules are present in the cell, and after several steps of synthesis reactions, adjusting, building, forming, making bonds, tryptophan will be made. So this, again, could be measured in an experiment, like how much of maybe some of these proteins are present, how much tryptophan is present. Do we have, tryptophan changing in quantities? This is obviously going to teeter and totter back and forth between the two of these. As more tryptophan is made, it's going to be available to bind to that inactive repressor and change it. So this is also another example of a negative feedback loop here, because if you're going to be able to regulate between the two, that's very efficient for a biological system. So for a reducible operon, here's its statement. Hey, this building block I need is already present in my cells. I better repress transcription of the genes that make the enzymes, which help to manufacture that building block. I don't want to waste energy. So for repressible operons, it's usually going to be ones that are synthesis reactions, are anabolic or building reactions. Remember that amino acids are used to build proteins, so we definitely need this building block to further other things. So we have to build it if we don't have it. When an amino acid tryptophan is already present in the bacteria cell, the cell does not need the tryp-making enzymes. So transcription of those genes is off or repressed. Okay, so for repressible operon, when X is present, operon is off. That's our repressible operon. Okay, or operon is blocked. G expression is blocked. Okay. I hope this is helpful for y'all. Again, the reason it's got the two sides is so you can kind of like read this information, flip it open, read that information, compare and contrast. There are some great resources that I've added to Schoology, so I'm going to attempt to get here to have my phone on my laptop. Right? That's fun. Whoop. How about I just bring laptop closer to phone? There we go. All right, so on Schoology, there is a couple of things in the online resources. There we go. I've got it all labeled as remote learning since school closure. So DNA and gene expression is our unit. And now we're down here to prokaryotic gene expression and operons. There is a PhET simulation if you would like to go through that one. So this is Gene Machine, the lac operon. You would just download and run it. It's safe for your computer. It's fine. Let me go ahead and see what it looks like and if it's quick. No, I don't want to do that. I want to play it. Okay, we'll just open it, see what happens. It should open up as a new tab. Okay. Oh, wonderful. Well, put a pin in that for me. I'll have to check if it works. I've uploaded some of the videos from Bozeman Science, Amoeba Sisters, and Khan Academy, and then my video will be right here under Ms.Weith. You've got the former stuff from last week, and if you're looking for the handout for the PhET simulator, it's here under handouts, optional digital learning while school is out, and now we're towards the bottom, okay? So stickleback fish was earlier this week, PhET Operon simulator, and once you kind of have gone through those, you'd be able to do worksheet 11 as well. If you want to click through all of this in the calendar, it's all there simultaneously. So we're here on operons, gene expression, video lecture, online sim, and then the various assignments that you can work on for March 27th, all right? Again, all of it's optional. You don't have to do it. Don't feel stressed if you're not. Any little bit is going to be helpful. So I'm going to be signing off to you guys, and I will also be putting up a AP Classroom formative assessment related to this week's material. So that will get unlocked today. Where am I at? Oh, it'll be here. I'll put it in. But just like there was an AP Classroom formative little check for last week's material, I'll put one up for this week's material as well, so that you can go about that as you like. All right. Talk to you soon, guys. Bye-bye.